Okay, welcome to the analysis of nanoparticles by Christopher Vassar.
Daniel Yanez.
And share my
What is a nanoparticle? A nanoparticle is defined by most labs as something very small, usually toxic, and encountered at the nanoscale. In industry, uh, it's 1 to 1,000 nanometers, and Food and Drug Administration is very specific because they accept 1 to 100 nanometers as a definition. The two studies we evaluated, we're calling the first and second study. The first is basically a safety study for dealing with nanoparticles, and the second was specifically concerning silver nanoparticles and E. coli and the response E. coli had to silver nanoparticles. Um, Fundamentally, they can be sourced from anywhere, given that these particles are engineered in the lab.
Now, moving on to the concerns and the reasons for the study. The safety of nanoparticles or the risk of exposure of tissue to nanoparticles is of interest for these industrial applications. Poorly soluble particles with low acute toxicity do impose dose-dependent chronic inflammation on these tissues. Surface chemistry is also an influence, which is non-homogeneous composition that can irritate and create cytotoxicity within these tissues. Mechanisms of action include pulmonary inflammation and fibrosis, some cancer risks, incomplete phagocytosis of asbestos nanoparticles are claimed to be the source of carcinogenicity of asbestos, and the interference with the function of organelles and other cellular structures.
review final question of the research article. We gathered that this question is it challenging for bacteria to evolve defenses to heavy metals? Silver nanoparticle resistance in E. coli represent a type of metal toxicity response. We gathered that this question is important in developing toxicity studies, and the evolutionary impact of nanoparticles on the bacteria themselves may be of interest. Knowing the genetic responses to toxicity at the level of bacteria can lead to important mediation strategies and applications such as environmental cleanup activities, as well as medical applications. For example, in the medical field, if bacteria can develop resistance, it threatens the effectiveness of nano-based antibacterial products. Furthermore, engineered nanoparticles may have unforeseen consequences when bacteria are forced to survive against such toxicities. Okay, based on the methods section of the article, we gathered this flow chart, and it begins with E. coli. The bacteria specifically used is E. coli K12 Mg1655, and it's cultured using a Davis minimal broth. The experimental groups include the E. coli that was exposed to silver nanoparticles and the control group that is not exposed to any nanoparticles. The experimental groups are exposed to silver nanoparticles at a concentration of 0.2 milligrams per liter. This is a sublethal amount, which means that it's not enough to kill all the bacteria, so it allows for resistance. This is repeated for 225 generations. During this, they measured the growth and the MIC. They measured growth using optical density to track the bacterial growth rates, and then they used MIC, which is the lowest nanoparticle concentration that prevented growth, to observe for resistance. They then tested for cross resistance. So the silver nitrate is a non-particle form of silver, and they tested resistance against this one, against the silver nitrate. The results actually showed that no resistance was specific to the resistance was specific to nanoparticles and not the ionic silver. Afterwards, they genome sequenced they use genome sequences to identify any mutations They used this to identify any mutations that were linked to the resistance, and their final gatherings show that the bacteria was indeed resistant to the nanoparticle.
So what was the key result? Basically that bacteria respond rapidly to nanoparticle exposure, and the and they basically evolve resistance to silver nanoparticles with relative ease. Here on the graph at the right, it shows an optical density over 24 hours at two silver nanoparticles concentrations, 500 micrograms per liter and 750 micrograms per liter. So controlled bacteria show no detectable growth at either nanoparticle concentration, and the treatment bacteria show strong growth. So basically, the bacteria that were expected to respond did. What did this mean to the researcher? That bacteria can evolve resistance to silver nanoparticles with relative ease, and because of that, Dr. Graves emphasized responsible use and monitoring of nanoparticles in consumer and medical products, because you never know what you're going to make.
Moving on to informative criteria that we obtained from these articles, we learned that nanoparticles, though small, can have huge environmental and biological effects. These nanoparticles, while they can be useful in a technological sense, can also be highly toxic to biological systems. There are different nano waste streams that are classified in classes based on how toxic they are to the environment and to us in general. Nanomaterials can cause specific damage to these cells and tissues. As mentioned in the article, it can cause pulmonary inflammation. Nanofibers can also enter cells without killing them and instead causing chromosomal missegregation, otherwise known as an error in the cell division. These nanomaterials could eventually be used to improve conditions in humans, such as removing toxins like metals from the blood. From the second article, we obtained that the effectiveness of nanoparticles against bacteria and viruses is due to their unique chemical and physical properties. The program BREC is what identifies the mutations in the genes in accordance to the sequencing of the genome. This increase in resistance to the treatment is not due to major genomic changes, as observed. Now, tying back these concepts to our course, the evolutionary toxicology of nanomaterials have influences to the fitness costs assessed in bacterial studies, as this one. The heavy metal exposure in bacteria can be associated with coevolution of antibiotic resistance. The distribution of microbiomes of larger organisms are of interest to the human scale. These model organisms are used to ascertain potential toxicity, longevity, fecundity, development, and the response to environmental stress and behaviors.
All of these things are relevant to our course. That's all for now. Thank you for listening. Please stay tuned to the next one.